kinase inhibitors are another important aspect of cancer therapy. And so the idea is pretty simple. It was noted early on that cell proliferation is under the control of the endocrine system, which means it's stimulated by growth factors and hormones. And growth factors basically bind to receptors on the surface of cells, and we talked about this kind of receptors, a long time ago when we discussed receptors. And in general have a extracellular component. So if this is the membrane, we have an extracellular component, a transmembrane component, and then an intracellular domain, which is a kinase, and so phosphorylated residues. And generally, these receptors work in concert, so they are dimeric, and activation by binding of one of these growth factors or hormones, in the case, for example, of estrogens, results in dimerization and phosphorylation of the other subunits, right? So with phosphorus. Okay, so we can target two different aspects in this process. We can target the extracellular domain, so that's a part of your assignment to figure out what monoclonal antibodies do, or we can target the intracellular pathways, which are dominated by kinases. So in general, these growth factors work by activating the RAS pathways. So we have again the growth factors bound to the receptors, phosphorylation, that in turns phosphorylated these proteins SHC, GRB2, and SOS, which contain a specific domain called SH2, which gets phosphorylated. Phosphorylation of these proteins in turns activated RAS, and again that's done through phosphorylation. And RAS has two main pathways. One pathway initiated by this P13K that leads to cell proliferation and, most importantly, inhibition of apoptosis. Another pathway, which has still been investigated, seems to increase the invasiveness and metastasis, so facilitates the getting out of the tumor and invading the vascular aspect. The most important pathway is the so-called MAPK, so mitogen activated protein kinase. And what it does is, again, leads to cell proliferation and also changes that are associated with the cancer cells. So what happens is that many cancers, RAS is out of control, either via mutations that make it constitutionally active, or we also, as we'll see, that can derive by constitutional activation of the receptors. And so the first situation in which we saw the possibilities of targeting tyrosine kinases and thus preventing cell proliferation is with the so-called Philadelphia chromosome. And so this was really the first case of targeted therapy for cancer, something that we hear a lot about in the news. This was done before we were able to sequence the DNA of cells in a economical fashion. And it was noted back in the 70s or in 60s rather, because scientists discovered an abnormality that was present in 95% of patients with chronic myelogenic leukemia. This abnormality is a translocation between chromosome 22 and 9, in which the ABL gene, which is originally on chromosome 9, gets translocated to chromosome 22. And in doing that, gets fused to BCR gene, which is a breakpoint cluster region. And that results in a specific diagnostic chromosome called the Philadelphia chromosome, which is a shorter chromosome 22. And so it turns out that the product of this gene is a tyrosine kinase that is always activated. And this altered tyrosine kinase phosphorylates and activates numerous pathways, including the MAPK and AKT-PKB, which we have seen. And so basically functions to activate things that are normally activated by a RAS. And that leads to proliferation of white cells, and normally lasts about a year, Um, and basically, this leukemia can be very lethal within a year. So, however, because this BCR-ABL fusion is characteristic of leukemia, allows us to have some specificity in targeting the leukemia. And so, as I said, normally this is a constitutionally activated, and so phosphorylated substrates and activate them. We can think of inhibitors, and imatinib, which is something that we'll discuss in a second, competitively inhibits this kinase, and so basically competes with the substrate and stops tumor cell proliferation. This is a structure of imatinib, which is the first highly specific kinase inhibitor that was developed specifically for CML, and it was developed actually initially from a screening of possible tyrosine kinase inhibitors. Initially, again, we didn't have the structure of the fusion protein, but then, once the structure was obtained, we were able to see the mechanism by which the inhibitor works. Let's look at the structure of this, really life-saving agent called Gleevec, which was developed specifically for this fusion product. It's an example of targeted therapy. It's now very expensive, and especially in the US, in countries where the National Health Service is nationalized, the price of Gleevec has been also negotiated, and it's much lower. It's very controversial that the price is so high because, again, one cannot think of a possible reason in terms of making this drug, and the current price is 10 to 30 times higher than the generic, and also has been calculated that the cost of the research to develop this cancer agent has been recouped. However, the patents are set to expire, so we'll see what happens next in terms of being able to afford this drug. This drug is very specific for CML, and again, 95% of the patients have this specific fusion protein, and so they will be responders. So with that, let's have another look at growth factors. So the success of imatinib led to other agents that could be used, in case of especially of resistance to this drug. The success of Gleevec also gave the idea that targeting tyrosine kinases could be extended to other situations, and for example, to the EGF receptor. This is epidermal growth factor receptor. So let's look at the biology of this receptor. It's a classic receptor that dimerizes upon binding to the ligand, which is EGF. And as the name indicates, this receptor is overexpressed on the surface of cells and leads to proliferation of epidermal cells. Let's see how it works in general. So again, binding activates the receptor, phosphorylating the cytoplasmic tail of each other. That in turn recruits SH2 and activates all the other proteins involved in the RAS pathways. And like we've seen before, there is also direct activation of the P13K and AKT pathway that leads to decrease in apoptosis, triggers angiogenesis, and therefore metastasis. And in general, this protein kinase it's all basically the messaging is carried out via phosphorylation. Okay, so how is that involved in cancer? So normally, this receptor is under the control of EGF ligand, and so it's activated by oligomerization, by dimerization, right? In a cancer, that regulation often disappears, and so sub members of this family, such HER1, HER2, HER3, and HER4 are overactive. And so being overactive can mean either being upregulated in expression, being overstimulated, which means they respond in an enhanced manner to the ligand, or it can be constitutively active. And so, for example, they can be constitutive dimers. These are also ideal drug target because we can block, as we see, at the 2 level, and that can be potentially very useful. So let's start by looking at protein kinase inhibitors, and those are gefitinib, which is on the market as Iressa, and erlotinib, which is in the market of Tarceva. Gefitinib targets EGFR, the tyrosine kinase domain. It's used in breast and lung cancer. Erlotinib works in chronic myelogenic leukemia. They have the same overall scaffold that you can see here, right? So clearly they bind to a similar target. This is very important because both receptors are involved in non-small cell lung cancers, which account for about 80% of all the cancers. And the EGFR is more expressed, is overexpressed in lung carcinoma tissue than it is in normal lung tissue.